Transcranial direct current stimulation, also known as TDCS, is a non-invasive neuron modulator that sends small electric shocks to the brain via electrodes in order to manually excite membranes from their resting potential. This in turn creates neuroplasticity and redirects neural pathways. Originating in the 1950s, has since been reborn and has been the phenomenon to change the fate of those with chronic depression, stroke victims, OCD, and even those with autism spectrum disorder, also known as ASD. It is able to excite and inhibit neurons within the cortex in order to stimulate and focus learning, memory, and motor functions. TDCS has moderate rates of success with little drawbacks. There are no known side effects, short or long term, and with repeated treatments can create greater possibilities and a progressive outcome for those with low cognitive functions. Transcranial direct current stimulation is used to stimulate the brain using small currents of electricity. Using TDCS does not directly induce cerebral activity. Instead, it non-invasively changes spontaneous activity within the brain as well as excitability by manipulating the subthreshold of neuronal membranes. By placing the electrodes in different locations on the head and using TDCS, it is possible to create neuronal activity in order to achieve specific outcomes. For example, by placing the electrodes on and around the prefrontal cortex, it is able to target higher order brain functions such as social skills, impulse control, reasoning, and mood. The anodes, positive electrical currents, are placed on one section of the head, and the cathode, negatively charged currents, are generally placed on the opposite side and used to send a small electrical current through the brain. By doing so, it creates a manual firing of the neurons simultaneously, entering via the anode, flowing through the brain, causing stimulation and excitation, then exiting through the cathode. By positively firing the neurons inside the brain simultaneously, it creates depolarization within the synapses of the cells, surpassing the threshold of excitability, which in turn opens the gates for calcium and magnesium to flow freely until it has reached resting potential again. Dependent on what is trying to be achieved and the location of the electrodes, the treatment is generally repeated for up to 20 minutes. By doing so, it creates neuroplasticity. Neuroplasticity being the changing of neural pathways due to the altering of neural processes into the direction of excitability. Studies have shown that using TDCS, it can improve one's focus, mood, and learning. Won Ma and Kim defined autism spectrum disorder, ASD, as a group of developmental disabilities characterized by impairments in social interaction, communication, and restricted and repetitive interests/behaviors. The left hemisphere is responsible for logic and reasoning. These qualities are generally not found within those who have ASD. Thus, when using TDCS, researchers place the positively charged anode on that side for neural firing. Also found in the brain of those with ASD are hyperplasia, amygdalas, and cerebellums, these being the locations that control emotion, memory, sensory, and motor functions. Allen and Co., 2003 and 2004, commented that in ASD, it is common that activation within the cerebellum area is significantly reduced when it comes to attention-focused tasks, although heightened when performing basic motor skills. As not all those with ASD are the same, some experience hyperplasia, whereas others experience a decrease in the size and neuron count within the amygdala. This could in turn be a leading cause as to why those with ASD struggle to read social cues and are generally more reserved than neurotypicals. To date, there is no treatment for autism, as it is a genetic and neuronal developmental disorder. TDCS does not offer that it can resolve ASD, rather shows promising results in decreasing some of the severity of the symptoms that correlate with ASD. Many pilot studies have been completed around individuals with ASD, and many of them present assuring effects. Due to its non-invasiveness, children are able to sit through treatments unharmed and at ease. Repetitiveness is key with children and in learning, especially those with ASD, and so the treatment with TDCS is exactly the same. By placing the anode, positively firing, over the left hemisphere, more specifically the prefrontal cortex, and the cathode, negatively firing, over the right hemisphere, a pathway of firing will form. It is hypothesized that TDCS could moderate neurons via depolarization and excitability within specific areas of the brain in order to lessen the severity of behavioral problems, communication issues, cognitive function, and motor skills. Osorio and Brunoni had discovered that their preliminary evidence is encouraging of the potential usefulness of TDCS for treatment of ASD in children and adolescents. It suggests tentative support for reductions in symptom severity and, according to parental reports and clinical observations, improvements in some aspects of language. Imatachaya and co. had found that post-treatment of TDCS led to a decrease in ASD symptoms and had lasted for up to 7 days based around the Childhood Autism Rating Scale, also known as CARS, specifically after using TDCS on the F3, which is the left frontal cortex. A lot of data found for this research was rather vague, as this is still a new area that is currently being explored. For TDCS to progress forwards in the area of ASD, there would need to be much more research conducted, not just in children but also in adults and adolescents, in order to gain a more comprehensive collection of data across the lifespan of those with ASD. It would need to be conducted in more ethnically inclined participants, as well as more gender-specific studies, as the ASD ratio is 4 males to every 1 female. As TDCS is on the rise again, there needs to be better developed and stronger guidelines as to define the ground rules of using TDCS on participants, especially children.